Buenas tardes, mi nombre es Juan Carlos Hernández Boluda y soy el presidente del Grupo Cooperativo GEMFIN. Os doy la bienvenida a la quinta reunión científica anual del Grupo GEMFIN, que tiene que ser en formato virtual, como todos sabéis. Nos hubiera gustado vernos las caras, pero sigue sin ser posible, pero esto también ha traído algunas ventajas colaterales, como por ejemplo la mayor difusión de algunas de las actividades del grupo. De hecho, en la reunión de esta tarde hay registradas más de 220 personas y además incluye una nutrida representación de Latinoamérica, personas a las cuales pues les doy también mi bienvenida. Aprovecho para recordar que estamos de enhorabuena en el grupo, es nuestro aniversario, seguramente que muchos no os habréis dado cuenta, pero llevamos 10 años desde la constitución del grupo, que se constituyó en 2011 en el Congreso de Zaragoza. Y estamos de enhorabuena. En este tiempo hemos formado un grupo muy ligado y muy productivo a nivel científico y reconocido más allá del país. Pues bien, vamos a empezar la sesión científica, que la hemos dividido en 2 días, en 2 días distintos para que pueda seguirse sin quedarnos demasiado fatigados en el intento, ¿no? La sesión de hoy tiene 2 bloques. Tiene un bloque de biología, que estará moderado por la doctora Cristina Bilbao, y luego una parte más de clínica, que será moderada por el doctor Jesús María Hernández Rivas. Y luego tendremos un pequeño capítulo nos hará algunos comentarios el presidente del grupo EMPI en España, y finalmente pues daré por concluida la reunión de hoy. Y mañana tendrá una parte de ponencias o simposios de la industria y luego pasaremos a la asamblea del grupo. La asamblea del grupo no es en abierto, sino que solo podrán entrar aquellos miembros del grupo GEMFIN. Me gustaría recordaros que todas estas charlas y los comentarios posteriores van a ser grabadas y la mayor parte de ellas podrán ser contempladas posteriormente en unos días en la web de GEMFIN por abierto. Me gustaría también, por supuesto, agradecer el apoyo económico de las compañías que han contribuido al mantenimiento del grupo y a la realización de esta sesión, fundamentalmente Novartis y BMS por su cuantía, pero también quisiera agradecer la colaboración de Insight y de STI Biopharma. Y por último, el soporte logístico de ENFAR, que de nuevo pues, ha sido estupendo y que nos ha permitido llevar a cabo esta interesante sesión. Pues, me gustaría que disfrutarais de las sesiones de hoy y ya sin más dilación paso el testigo a la doctora Cristina Bilbao para que modere el primer bloque del día de hoy. Un saludo.
Buenas tardes, mi nombre es Cristina Bilbao y trabajo en el servicio de hematología del Hospital Dr. Negrín de Las Palmas. Voy a moderar el primer bloque de biología, que creo que va a ser muy interesante por el alto perfil que tienen los ponentes, y recordaros que podéis hacer preguntas a los ponentes a través de la plataforma, a través del canal. Me voy a pasar al inglés para introducir a los ponentes. Good afternoon, my name is Cristina Bilbao. I work for the Hematology Department en el Hospital Dr. Negrín de Las Palmas, and I will moderate the biology session. I'm very pleased to introduce our next 3 speakers. I'm very happy they have accepted our proposal to participate in the annual GEMFIN meeting and talk to us about the recent advances and their lines, as they are highly acknowledged researchers. The first speaker is Dr. Nangalia, a clinician scientist from the Cambridge Stem Cell Institute in UK, and she would talk about in utero acquisition of driver mutations in patients with MPN. Our second speaker is Dr. V. Sankaran from the Boston Children's Hospital, Harvard Medical School in Boston, and he will talk about inherited myeloproliferative neoplasm risk affects hematopoietic stem cells. And the third speaker is Doctora Marta Garrote del Hospital Clínic de Barcelona, que presentará, and she will present the study, Estudio Molecular de los Pacientes con Trombosis Esplácnica. Thank you very much to the 3 of them.
Thanks so much for the invitation to speak today. It's a real privilege. I'm looking forward to telling you about our recent work where we find that early driver mutation acquisition and lifelong clonal expansion underlie adult myeloproliferative neoplasms. So the motivation for this work was questions that are commonly posed by our patients to us as clinicians when they come for their diagnosis. They ask, how long have I had this for? How fast did it grow, and is it still growing? And the myeloproliferative neoplasms are a real unique model within which we can address this question. There are three flavors of MPN, as you all know: polycythemia vera, essential thrombocythemia, and myelofibrosis. And this audience needs no introduction into the various phenotypes, but we all know that these are chronic cancers that affect patients for many decades, so we can sample patients' blood and study them over that time period. Also, because they are quite simple at the genomic level, they give us a window into the earliest stages of cancer development, and some patients often progress genomically and phenotypically to myelofibrosis and acute leukemia. So we have the ability to model the disease progression in these patients as well. And importantly, these clones, these mutant clones that cause MPN, exist alongside normal hematopoiesis. And so we can compare what's happening in the mutant clone to what's happening under the normal situation in the patient. Now, in order to time the driving mutations, we need to know what we're looking for, and MPNs are a great model because they're very well genomically characterized. 90% of patients have a mutation in JAK2, calreticulin, or MPL, and 60% of patients have mutations in additional myeloid cancer-associated driver genes, such as TET2, DNMP3A, and also many patients have chromosome 9 aberrations. This leads to homozygosity for mutant JAK2. So our experimental design involved 10 patients aged and diagnosed between the ages of 20 and 76 years. We studied patients with ET, PV, and MF. And in these patients, we took their peripheral blood or their bone marrow samples, and we grew single-cell derived colonies from individual cells. Now, each colony underwent whole genome sequencing. In total, we had around 1,000 colonies that we underwent whole genome sequencing for. And then what we did is use the mutations present in each individual colony and compare them across all the colonies in individual patients to draw a family tree of somatic mutations. And that is because right from when we are a zygote, dividing into 2 to 4 to 8 cells, every cell division and the passage of time leads to our cells acquiring mutations. And over time, if we then take a snapshot of the mutations present in individual cells, this allows us to trace back the history of those cells back to birth. So we did this in our patients with MPN. To start with, we first called the driver mutations in all these individual colonies, those 1,000 colonies in our 10 patients, and we found the usual players involved in terms of mutations, but at the clonal level, we found very many more driver mutations present in colonies than was apparent from bulk whole exome sequencing, showing that in individual patients, there are many more driver mutations present at very low levels that would only be detectable if you look at the single cell level. And then what we did is use somatic mutations to essentially build a family tree of hematopoiesis. And what we're doing there is drawing a tree where the branches represent mutations. So say we have colony A and colony B. Let's have this. If we have colony A and colony B, the mutations that are unique to colony B or colony A are in the end branches, and mutations that are shared between colony A and B go in a higher branch and thereafter, such that mutations at the top of the tree are shared by the majority of colonies and occurred early in life, and mutations at the bottom of the tree occurred later in life. What we can then do is find out which colonies have the driving mutations and which colonies don't, and time the driving mutations to the branches of the tree. So here we can say that the green driver mutation occurred before the orange, which then occurred before the blue driver mutation. But we can go one step further. We can convert a mutation tree to a time tree. And that's because we know that somatic mutations in our genome occur constantly over life. All of our individual stem cells in blood are acquiring roughly 18 mutations per year, which then allows us to put an age at the start and the end of each branch, and thereby give us an estimate within which these driver mutations occurred. And that's what we did. So let's move on to our first tree. This is a patient diagnosed with ET at a relatively young age of 21 years, presenting with an asymptomatic thrombocytosis. And in this patient, each individual red dot is an individual colony that underwent whole genome sequencing. And we can see mutations along the y-axis. So all the colonies have on average 500 mutations, which is what is expected for a 21-year-old. On the left-hand side, we can see wild-type hematopoiesis. Wild-type hematopoiesis diverges very early in life. They don't share very many mutations amongst them. And this tells us that the stem cells that are producing our normal blood diverged early in life and have been independently producing blood for most of our life. However, on the right side of the tree, you can see colonies that have diverged later in life. They've diverged around 80 mutations of mutation time. And these colonies all share JAK2, which localizes to a branch that we time to between 6 weeks post-conception in utero to at the very latest 1.3 years of standard age, telling us that this JAK2 mutation was acquired 20 years before this patient presented with MPN. The pattern of branching thereafter tells us how quickly that single stem cell expanded into a clone of stem cells to then contribute to mutant hematopoiesis over life. In the second patient diagnosed with PV at the age of 31 years, we see a very similar picture. In the middle of the tree is the MPN clone mutant for JAK2. And again, JAK2 here is acquired in childhood, at the very latest by 8.5 years of age, in a patient diagnosed with PV several decades later. But what's also interesting is this patient has an independent DNMT3A mutated clone. So DNMT3A is one of the commonest mutations in age-related clonal hematopoiesis, but this young patient's clonal hematopoiesis clone acquired a DNMT3A mutation by age 7 to 8 years of age, which is quite unusual. In some patients, we have a very complex picture. In this patient presenting with PV at age 33, this patient actually presents with Budd-Chiari syndrome. There's a complex driver mutation landscape with many mutant HSCs contributing to blood production. Indeed, in this patient, JAK2 was acquired very early in life in utero, and indeed their DNMT3A mutation was also acquired in utero. Indeed, we found other patients where there was very early DNMT3A mutation within a few weeks of life, telling us that these driver mutations can often occur very early in life, at least in our MPN patients. What we also then see is there's recurrent driver mutation acquisition within the mutant clone over life. So by age 17 years, this JAK2 clone became homozygous. And then by age 33 years, this JAK2 clone developed amplification of chromosome 1q+, which is often associated with transformation of disease. Now, in some patients, JAK2 is acquired second. In these two patients diagnosed later in life, in their 50s and 70s with ET, JAK2 occurs as a second mutation on a clonal expansion that has a DNMT3A mutation. What's particularly interesting is that in this first patient, they had R882H, the most commonest DNMT3A mutation, occur within childhood. That clonal expansion is then sitting there waiting to acquire a second driver mutation that then, in this case, because it's JAK2, leads to the MPN phenotype. And in some patients, the most elderly patient in the cohort, we found clonal expansions within the wild-type compartment, perhaps suggesting that there are driver mutations as yet to be uncovered in clonal hematopoiesis. Now, in some patients, a recurrent theme we found is that the same driver mutation is being acquired multiple times in the same patient. So in this patient who was diagnosed with PV at age 53 with an exon 12 mutation, again, acquired in childhood by age 9, the patient then progressed to MF later in life. This patient has 3 different DNMT3A mutations and 4 independent acquisitions of homozygosity for their exon 12 JAK2 mutation, suggesting that there is a predisposition to getting the same events multiple times, maybe because those driver mutations are under selection in that patient. Now, what about MPNs lacking phenotypic driver mutations? We know that 10% of ET patients lack CALR, MPL, or JAK2 mutations. So we've recently looked at triple negative MPNs. This patient here PD6634, presented with ET at age 27 with a platelet count of nearly 2,000. And they are now in their 60s and have been on hydroxycarbamide for many years. And we can see that this patient, most of their blood is mutant for a PPM1D mutation, which is typically associated with clonal hematopoiesis. But of course, this PPM1D mutation was again acquired in childhood. In the second patient, who was actually diagnosed with PV, JAK2 negative, but confirmed by a raised red cell mass, and is now in their 90s, also on hydroxycarbamide, the vast majority of their blood is double mutated for PPM1D and TET2. Now, how these clones are determining the phenotype of MPN, we don't know whether they are just bystander clones or in some way driving the phenotype of MPN. We then went to ask ourselves the question, okay, we know when these driving mutations are occurring, how fast are they growing? So we went back to our patients and we took blood samples again, bulk blood samples this time, and within those bulk blood samples, we looked for the mutations that were present on our phylogenetic tree. And by looking at the pattern of branching of the tree and the clonal fractions in bulk blood, we were able to infer the historical average growth rates of the clones since the time of acquisition. So what we could then do is put a number on the rate of additional growth per year. And what we found is that in the same patient with multiple different clones, the clones are growing at different rates. In this individual that has an in utero acquired DNMT3A mutation, it's growing very modestly at less than 10% per year, taking 40 years to reach a 1% VAF or clonal fraction in the blood. Whereas some of their other MPN-related clones, their JAK2 homozygous clone and their JAK2 and TET2 mutant clones are growing very fast. 233% a year means the clone is doubling in size every 7 months. And in some patients that just had a JAK2 mutation, we found that the JAK2 clone is growing at different rates in different individuals, suggesting that it is not just JAK2 that determines the rate of growth of the clone, but other factors, maybe inflammation or the germline predisposition of the individual, that determines the consequence of JAK2 mutations in an individual patient. We then asked ourselves, well, if we know that these mutations are occurring early and we know how fast they're growing, would we have been able to detect them early in these individuals? Well, for the slowest growing clones, we think with sensitive assays, we would have been able to detect the clone up to 40 years before diagnosis. And for our most fast growing clones in the context of disease progression, we may have had a 10-year head start in detecting the clones in bulk blood using sensitive assays. And this then begs us the question, can we then predict who's going to suffer from these rapid clonal expansions? If we could detect early, could we identify which patients are going to be the fast growers and which patients are going to be the slow growers? And that we found strongly correlates with latency to disease presentation. So what we can now hypothesize is that if we can determine the growth rates of the clones at an early time point, then we can determine the latency to disease on this y-axis by plotting individual patients. So in summary, I've shown that in utero and childhood driver mutation acquisition for JAK2, DNMT3A, and now PPM1D have been identified in patients that develop MPNs. These clones can often acquire new driver mutations over the lifetime of patients, often separated by decades. There are variable rates of clonal expansion, which could be a combination of both the driver mutation and patient effects. Importantly, expansion rates determine latency to diagnosis. And my view is that if we could detect these mutations early and determine their rates of expansion, we could possibly predict those patients or individuals in the healthy population on a path to clinical manifestations that could enable preventative strategies. I'd very much like to thank the first author on this study, Nick Williams, the members of my group, my mentors, Peter Campbell and Tony Green, and our other collaborators, as well as our funding organizations, Cancer Research UK, Wellcome, the MPN Research Foundation, and EHA, our patients who supplied their precious samples to allow us to study them in detail. Our full paper is on bioRxiv, as well as on our Twitter profile. Thank you very much. I'd be happy to take any questions.
It's an incredible pleasure to be able to present at this GEMFIN meeting. I wanted to today talk about some of our work on how inherited factors can affect the risk of acquiring myeloproliferative neoplasms and how we can better understand the underlying mechanisms of these variants. Over the last several years, as you heard about in the prior talk, there's been an incredible amount of work to define the somatic mutations that cause and drive myeloproliferative neoplasms. We now have a near comprehensive understanding of these driver mutations. At the same time, it's important to bear in mind that myeloproliferative neoplasms have a significant heritable risk. First-degree relatives of individuals who have myeloproliferative disorders are at 5 to 7-fold increased risk of themselves acquiring a myeloproliferative disorder compared to the general population. This is nicely illustrated here in the Swedish population registry, where you can see that with age, there's an increased incidence of myeloproliferative neoplasms in those with first-degree relatives. This is a higher familial risk than most other adult cancers, including breast cancer, prostate cancer, and colorectal cancer. So there's been work in the past to identify a number of variants, including a haplotype of the JAK2 allele, as well as a handful of other variants. However, there's been a number of questions that have remained unanswered. First, what other variants might underlie this risk? And second, what are the underlying mechanisms by which these variants can confer increased risk of acquiring myeloproliferative neoplasms? The work I'll be describing was led by Eric Bao, Satish Nandakumar, and Xiaotian Liu in our lab. We initially started off by conducting a genome-wide association study for approximately 3,000 individuals with myeloproliferative neoplasm in a total population of 1.1 million individuals. By performing this analysis, we could identify 25 genome-wide significant or suggestive loci, including 17 that were genome-wide significant. This revealed a variety of loci, and I'm just showing you here in this Manhattan plot the nearest genes, as well as the location of these different variants. As we developed a polygenic risk score by using these variants, we could see that those individuals at the top decile in an out-of-sample population actually had about a threefold increased risk compared to the median deciles, and a nearly 4-fold increased risk compared to the bottom deciles. This suggested that the variants we were identifying in aggregate have a significant risk of conferring predisposition to myeloproliferative neoplasms. Now, we then wanted to ask, what in vivo phenotypes could we use to gain mechanistic insights, and how might these variants be acting to confer increased risk? We started our study by looking at blood cell traits, which we frequently study. As you're all aware, blood-forming stem and progenitor cells are able to produce all of these lineages, as I'm showing you here. And when we looked at a genetic correlation between those genetic variants that we identified through our genome-wide association study of factors that are in the germline predisposing individuals to myeloproliferative neoplasms and genetic variants associated with a number of blood cell phenotypes, we could see that across a range of blood cell phenotypes representing all of these distinct lineages, we could see largely positive correlations, as I'm showing you here. The only outlier was mean reticulocyte volume, which showed a negative correlation, but indeed red cell count showed a positive correlation. This suggested that across a range of traits, there was a positive correlation with blood traits, suggesting that these variants might be acting at the level of multipotential stem or progenitor cells. We then wanted to gain further insights. One measure of stem cell activity is through telomere length. We know that as stem cells maintain their telomere length when they self-renew, but then as they undergo subsequent differentiation, the cells will then have attrition of telomere length. Variation in telomere length can give a metric of variation in stem cell number within the blood-forming compartment. We were also motivated by the significant dissociation we had noted at the TERT locus, as I'm showing you here, on chromosome 4. This suggested to us that perhaps telomere length might be having an important role. We decided to use as an instrument to test this potential factor, leukocyte telomere length genome-wide association studies from approximately 80,000 individuals. By looking at a correlation at the TERT locus specifically between the effect on telomere length of any particular variant and the effect upon MPN risk, we can see that there was a positive correlation as shown here, where those variants that increased telomere length were associated with increased risk of acquiring MPN. However, we wanted to look across the entire genome, not just at a single locus. So we could do this by performing a Mendelian randomization, where we treated the effect of any particular SNP on telomere length as the instrument, and then looked at the effect on MPN. And indeed, what we could see across a variety of Mendelian randomization approaches, all of which have different assumptions and underlying methods, we could see that there was actually a positive genetic correlation, irrespective of which method we used, including MR-EGR, IVW, and weighted median Mendelian randomization approaches. This demonstrated that variants that have an effect on telomere length were having a direct and causal impact on MPN risk. Of course, this could be mediated through upstream factors. For instance, factors that affect stem cell self-renewal might affect telomere length, and then in turn would also impact MPN risk as well. These in vivo phenotypes suggest a role for stem cells in this phenotype, but we wanted to get at this using independent approaches. To do this, we initially examined accessible chromatin data across the hematopoietic hierarchy. Well, we looked at the most significant variants and asked which variants and which cell states do these variants map to. We could actually see that the most accessible chromatin was located within blood-forming stem cells and multipotential progenitors that are immediately downstream of these stem cells, whereas other blood cell progenitors and differentiated cells showed little to no enrichment. This suggested that many of the variants were acting at the level of the blood-forming stem cells or multipotential progenitors. We could also take the same accessible chromatin data and look across all associated variants using the LD score regression approach. And once again, we got a very similar result, where now taking into account not just the most significant loci, the 25 that I had mentioned before, but all the genome-wide significant, or all the genome-wide signal that we had, we could see that there was a high enrichment in stem cells and multipotential progenitors, as well as lower degrees of enrichment across other progenitors and differentiated cell types. Once again, this highlighted the key role that stem cells were likely having in the underlying predisposition to myeloproliferative neoplasms. We then wanted to take approaches to try to nominate potential target genes of all the variants we had identified. This could include nominating genes that contained coding variants that were nominated through this GWAS. But in the majority of instances, we identified non-coding regions of the genome, and we could then start to nominate genes either in which the non-coding variants were contained, or genes that might be connected through enhancer to promoter connections to different genes, either using promoter capture high C or correlations between gene expression and accessible chromatin. Using this approach, we could identify a series of genes, as I'm showing you here. Interestingly, the majority of these genes are factors that are well characterized and important for stem cell self-renewal and function. And indeed, the majority of these genes are very well expressed in blood-forming stem cells. Well, what I've shown you so far is that our global analyses can nominate stem cells as underlying the risk for predisposition to myeloproliferative neoplasms. But I want to give you an example of how we can start to perform targeted variant to function analyses to gain further insights. At one particular risk locus, there was this region downstream of the gene GFI1B that contained a couple of risk variants. These map to a region of accessible chromatin in blood-forming stem cells and downstream progenitors. When we took this region out of the genome and asked, does it have any transcriptional activity, we could indeed see that one of these variant-containing regions contained robust activity, as I'm showing you here, RS524137. When we then looked at the alternative alleles using this reporter assay, what we could see is that the risk allele was associated with reduced enhancer activity, suggesting that this was associated with reduced regulation of this regulatory element. However, we then wanted to look at the endogenous activity in blood-forming stem cells. We could do this by performing CRISPR-Cas9 genome editing in blood-forming stem and progenitor cells. We could edit approximately 60% of the cells in bulk by using dual guides flanking the regulatory element. Using this approach, we could reduce GFI1b expression by approximately 30% in both the bulk stem and progenitor cell population, as well as in the phenotypic long-term hematopoietic stem cell population. Importantly, if we then took these cells out further in culture, what we could see is by about 7 days in culture, we had an expansion of phenotypic long-term stem cells. And indeed, this expansion was selective to the long-term stem cell compartment, and there was not nearly the same degree of expansion seen in other compartments, including the bulk CD34 positive compartment or the entirety of all cells. We also performed colony replating assays, where what we could do is plate cells at low density in methylcellulose, semisolid media, and then attempt to look at the phenotype. If we disrupt GFI1b, the coding region at least, we can get rid of or nearly eliminate erythroid colonies as shown here. However, these cells have a propensity to self-renew, and if we secondarily plate the colonies, we can get increased self-renewal. In contrast, by deleting the enhancer, we actually see no suppression and in fact an expansion in primary colonies, including of erythroid colonies. But interestingly, we also get increased secondary replating as a result, suggesting once again that this regulatory region harboring a pathogenic risk variant is conferring increased self-renewal upon stem cells and thereby resulting in increased risk for myeloproliferative neoplasms. So in summary then, the model that emerges from the work that I've shown you today and additional work that we have performed that I didn't have time to discuss is that there's variation in germline genetic risk, with some individuals having some degree of self-renewal and being at lower risk, although those individuals can still continue to acquire somatic mutations such as JAK2 V617F and progress to get an MPN. And then there are individuals at higher germline risk who seem to have more self-renewal, and this then causes an increased risk of progression to MPNs in those individuals, as I demonstrated. Importantly, these findings not only give us insights into the factors that put individuals at risk for acquiring a myeloproliferative neoplasm, but they also suggest avenues by which, once we understand those who might be at genetic risk, how we might be able to mitigate that. For instance, increased levels of ascorbic acid or vitamin C have been shown to enable and reduce self-renewal of stem cells, and using such a strategy, particularly those at risk, might help mitigate this risk. With that, I wanted to finish my talk by acknowledging those involved in the work I've had a chance to discuss today, including Eric Bao, Satish Nandakumar, Xiaotian Liu, and other members of our lab and collaborators, along with our funding sources. And I wanted to thank you for your attention today. Thank you very much.
Bueno, buenas tardes. En primer lugar, quiero agradecer a GEMFIN la oportunidad para poder dar esta charla en la que voy a hablar de las neoplasias mieloproliferativas con trombosis esplácnica y dar una visión global. Es conocido que las neoplasias mieloproliferativas tienen un riesgo incrementado de trombosis, que esta trombosis puede afectar a sitios atípicos, incluyendo el territorio esplácnico. Las trombosis esplácnicas en población general, las no cirróticas no tumorales, son acontecimientos muy poco habituales y se clasifican en función del territorio anatómico afecto. En el síndrome de Budd-Chiari, que afecta a las venas suprahepáticas y que es la entidad menos frecuente, la trombosis portal y luego las trombosis esplénica y mesentérica. Es conocido que la mutación de JAK2V617F es una causa principal de trombosis esplácnica, que de hecho en las trombosis esplácnicas no cirróticas no tumorales está presente en aproximadamente el 20% de los pacientes y que la trombosis esplácnica, a pesar de ser eventos poco habituales en la población general, se encuentra entre un 1 y un 10% de los pacientes con neoplasias mieloproliferativas. En relación a las neoplasias mieloproliferativas con trombosis esplácnica, se sabe que alrededor del 60% de los casos la trombosis es la primera manifestación de la enfermedad, mientras que en un 40% de los pacientes esta trombosis ocurre de forma evolutiva. En la mayor parte de casos, alrededor de un 70%, se trata de trombosis portales y en un 30% de los casos se trata del síndrome de Budd-Chiari. En un porcentaje no despreciable de pacientes hay una presencia de más de una trombosis en este territorio. Se ha postulado y cada vez hay más trabajos en los que parece que está claro que las neoplasias mieloproliferativas que debutan con trombosis esplácnica tienen un perfil demográfico y unas características fenotípicas específicas, siendo estos pacientes más jóvenes, con un predominio de mujeres y predominio de policitemia vera, y con una menor expresión hematológica, con hasta un 10% de los casos que no se pueden clasificar en ninguna de las categorías de neoplasia mieloproliferativa y con una mayor prevalencia de trombofilias si lo comparamos con las neoplasias mieloproliferativas no seleccionadas. En este sentido, dentro del grupo GEMFIN se publicó el año pasado este trabajo en el que se intenta ver un poco la historia natural de los pacientes que debutan con trombosis esplácnica. En este trabajo se analizaron 118 pacientes con policitemia vera o trombocitemia esencial que debutaron con trombosis esplácnica, se excluyeron las mielofibrosis y también en este caso, en este trabajo, las trombosis evolutivas quedaron como controles. Aquí vemos las características clínicas de los casos frente a los controles en este trabajo mencionado. Vemos que claramente los casos tienen una mediana de edad significativamente más baja. Por este motivo, probablemente hay una menor prevalencia de factores de riesgo cardiovascular y también vemos que hay una menor expresión analítica. En relación al tipo de neoplasia mieloproliferativa que presentaban los pacientes, vemos que los casos están enriquecidos con policitemia vera y que un 5% de estos pacientes quedaban como neoplasia mieloproliferativa inclasificable y que esto era muy poco frecuente en el grupo control. En relación al predominio del sexo femenino, en el estudio de GEMFIN esto no se vio que fuera estadísticamente significativo y esto se ha estudiado en otros trabajos y si bien es verdad, parece que no está claro que hay un predominio del sexo femenino en este subgrupo de pacientes, sí que parece que en el subgrupo de policitemia vera con trombosis esplácnica sí que hay una mayor proporción de mujeres. En relación a las causas de muerte en este subgrupo de pacientes, en el trabajo de GEMFIN se vio que los pacientes que debutaban con trombosis esplácnica morían de forma más frecuente por complicaciones relacionadas con la misma trombosis, es decir, por hepatopatía, por sangrados mayores o por segundas neoplasias, mientras que la muerte relacionada con la progresión de la enfermedad era un fenómeno poco habitual en el grupo de casos. Aquí vemos en este gráfico que no hubo ninguna muerte por progresión a leucemia mieloide aguda en el grupo de los casos. La supervivencia también se ha estudiado en varios trabajos en este subgrupo de pacientes y se ha encontrado en varios trabajos que probablemente también en el trabajo de GEMFIN no hay diferencias en la media de supervivencia si lo comparas con un grupo de neoplasias mieloproliferativas no seleccionadas. Pero como hemos comentado que estos pacientes tienen unas características demográficas diferentes, en este trabajo de GEMFIN se hizo por primera vez lo de ajustar la supervivencia por sexo y edad, y haciendo este ajuste, pues sí que se vio una reducción en 5 años en la expectativa de vida, y que esto fue estadísticamente significativo, por lo que los pacientes con trombosis esplácnica, comparados adecuadamente, tenían una peor supervivencia. En relación al riesgo de futuras trombosis y sangrados en este subgrupo de pacientes, sí que se sabe que este grupo de pacientes tienen una mayor incidencia de trombosis venosas, que principalmente afectan al área esplácnica. De hecho, entre un 10 y un 20% de los casos tendrán recurrencia de la trombosis esplácnica. En cambio, la incidencia de trombosis arteriales es igual o inferior si lo comparamos con mieloproliferativos no seleccionados, y que estos pacientes sí que presentan un mayor riesgo de sangrado, pero que principalmente está relacionado con la hepatopatía y el desarrollo de varices esofágicas. Y lo que sí que parece es que estos pacientes no tienen mayor riesgo de sangrado en otros territorios. En relación a la mutación de JAK2 V617F, se sabe que este subgrupo de pacientes está enriquecido con la presencia de esta mutación. En el trabajo de GEMFIN se vio que el 97% de los casos presentaban esta mutación frente al 87% de casos en controles, y estos resultados son consistentes con lo que se ha visto en otros estudios. A pesar de que se ha postulado de que la carga alélica de JAK2 es más baja en estos pacientes porque son más jóvenes o porque se encuentran en estadios más iniciales de la enfermedad, esto es controvertido y hay información discordante entre los varios trabajos que hay publicados. En este sentido, creemos que es necesario que se hagan más estudios para dilucidar esta cuestión, y teniendo en cuenta el ajuste por edad, la cronología de la trombosis y también aspectos técnicos de cómo se realiza esta determinación. En relación al perfil genómico de este subgrupo de pacientes, más allá de la mutación driver, es muy limitada. Y de hecho, hasta la fecha, solo hay un trabajo publicado que mire, en nuestro conocimiento, que mire específicamente el perfil molecular y la clasificación de riesgo en los pacientes con neoplasia mieloproliferativa y trombosis esplácnica, que se publicó este año pasado. Y en este trabajo incluyeron a 80 pacientes con mieloproliferativo y trombosis esplácnica, y que estaban divididos, que estaban repartidos en los 3 grupos cronológicos, siendo la mayor parte de ellos, un 80%, pacientes en que presentaban la neoplasia mieloproliferativa y la trombosis esplácnica de forma concurrente. Para hacer el perfil genómico de estos pacientes, utilizaron un panel de NGS comercial en el que se incluían a 36 genes mieloides, y lo que miraron en este grupo de pacientes es cuántos de ellos progresaban a mielofibrosis, a leucemia aguda o morían. Y lo que la principal hallazgo de este trabajo era que un 13% de estos pacientes presentaban la progresión o muerte, y que esta progresión o muerte estaba asociada con cargas alélicas de JAK2 por encima del 50% y mutaciones adicionales afectando a genes de la cromatina o del splicing. Esta tabla está sacada de la publicación de este trabajo, en la que vemos que en casi todos los pacientes la mutación driver era la de JAK2 V617F, y que en casi la mitad de los pacientes había otra mutación adicional, siendo las más frecuentes mutaciones en TET2, DNMT3A y ASXL1. A partir de estos datos y dado que hay poca información en relación al perfil genómico, en nuestro grupo hemos empezado a revisar el perfil genómico de los pacientes con neoplasias mioproliferativas y trombosis esplácnica. De momento tenemos analizados a 92 pacientes que también los hemos dividido según la cronología de la trombosis. Vemos que en la serie que de momento hemos analizado, un 60% de los pacientes tienen los diagnósticos concurrentes. El análisis genómico también lo hemos hecho mediante técnicas de NGS, en la que hemos usado un panel comercial con la presencia de 30 genes mieloides. A partir de los datos genómicos de los pacientes, lo que hemos hecho es clasificarlos según la clasificación genómica propuesta por Greenfeld en 2018. Y estos son nuestros resultados, en los que vemos que a la mayor parte de los pacientes, un 60%, caen en la categoría de mutación de JAK2 en heterocigosis, y que la segunda categoría más frecuente es la de mutaciones en genes de la cromatina y del splicing, en que hay un 17% de los pacientes. Cuando se hace un análisis por subgrupos según la cronología de la trombosis, lo que vemos es que aquellos pacientes que presentan la trombosis previa o concurrente son grupos que están enriquecidos con la mutación de JAK2 en heterocigosis, y de hecho en esta tabla vemos claramente que la mediana de carga alélica de JAK2 es superior en el grupo en que la trombosis es evolutiva si lo comparamos con los otros dos grupos. Las mutaciones adicionales más frecuentes que encontramos fueron en TET2, ASXL1 y DNMT3A, sin diferencias entre los grupos, y vemos que también hubo diferencias en la mediana del número de mutaciones que encontramos, siendo superior en el grupo de la trombosis evolutiva. A partir de esto, lo que hicimos es, como he comentado, pues la clasificación genómica según la cronología de la trombosis esplácnica, y en este gráfico de aquí vemos que los grupos de trombosis previa o concurrente están enriquecidos o hay un predominio de la categoría de JAK2 en heterocigosis, mientras que los pacientes con trombosis evolutiva hay una mayor representación de pacientes con TP53 o mutaciones en la cromatina o el splicing, es decir, en grupos de alto riesgo. De estos datos, lo que podemos concluir es que los pacientes que debutan con la trombosis esplácnica tienen menor complejidad genómica, y que esto, junto con la hepatopatía y la hemodilución, podría contribuir a que estos pacientes tengan un cuadro hematológico menos expresivo. Con toda esta información, pues nosotros consideramos que además de mirar el estudio de la trombofilia en los pacientes que tienen una trombosis esplácnica, se debe hacer un screening con la determinación de la mutación de JAK2 V617F en todos estos pacientes, y que además, especialmente en los casos de síndrome de Budd-Chiari, esto seguramente sería interesante complementarlo con un análisis genómico mediante NGS. El tratamiento, el abordaje terapéutico de estos pacientes sigue siendo controvertido, aunque sí es verdad que en la mayor parte de casos se recomienda tratamiento con antagonistas de la vitamina K de forma indefinida, y recordar pues, que son pacientes que son complejos y que se requiere un abordaje multidisciplinar. Ya para concluir, decir que la mutación de JAK2 V617F es una causa principal de trombosis esplácnica, que entre un 1 y un 10% de las neoplasias mioproliferativas presentarán una trombosis esplácnica que puede aparecer en diferentes puntos cronológicos de la enfermedad, que los pacientes con mieloproliferativo y trombosis esplácnica tienen un perfil demográfico y unas características fenotípicas diferenciadas, siendo pacientes jóvenes, con predominio de policitemia vera, menor expresión analítica, y que probablemente los pacientes con este subgrupo de pacientes tienen un perfil genómico diferenciado, con una prevalencia de la mutación de JAK2, con una carga alélica menor y menor complejidad genómica, y que el riesgo de progresión de la enfermedad es muy bajo y que la supervivencia principalmente está relacionada con la enfermedad hepática, y que clasificar genómicamente estos pacientes sería importante para establecer e identificar en este subgrupo los pacientes de alto riesgo. Y ya está. Gracias.
Congratulations to all the speakers for your amazing talks. They have been very, very interesting. And we have here some questions. The first one is for Dr. Nangalia. And the question is, do the mutated JAK2 clones acquire mutation faster than the wild type ones? And could this alter the estimation of the age of acquisition?
Yes, that's a very good question. So we find in some of our patients that the mutated colonies do have slightly more increased number of mutations, but we're talking very modest. We're talking a few extra mutations per year. Now, when we model the increase in cell divisions that have occurred in order for that clone to go from one stem cell to a clone of stem cells, the increase in mutation burden that we see is fully in line with the increased number of cell divisions that you predict for that clone to have. So we also find that the telomeres have shortened in those mutated colonies, which also suggests that this is due to cell division. So in answer to your question, we do see a mild increase in mutation burden. This would not affect timing because it happens after the driver mutation is acquired rather than before. And so up until the point at which the driver mutation is acquired, the rates between wild type and mutant would be the same.
Okay, thank you very much. I have a question for Dr. Nangalia too. Do you believe, or do you know if people with clonal hematopoiesis also develop the mutations in uterus?
So we don't know the timing of acquisition of JAK2 in clonal hematopoiesis. However, there are some papers that have modeled the rate of growth of JAK2 in the healthy population. There was a paper by Jamie Blundell's group published in 2020 Science, and that models the rate of growth of JAK2 to be even slower in the healthy population than in MPN patients. So if you imagine that in MPN patients, JAK2 acquires early and is growing faster, then for it to be growing slower in the healthy population, one can imagine that it may have also occurred at a similar time for it to then grow at a slower rate and then still be detectable in the elderly population. I wouldn't be surprised if it was similar childhood, just because these clones take such a long time to manifest, even when they're growing fast. Whether specifically MPN patients are at risk of in utero acquisition, the super early, I don't know. We certainly see even DNMT3A mutations acquired in utero in the MPN patients, and time will tell when we do similar studies in patients with clonal hematopoiesis, whether there is a difference in timing that then leads for one patient, purely because they've acquired it early, for them to have sufficient time to develop an MPN. So there is a possibility that it's not just the difference in growth rates between the healthy and the MPN patients, but also maybe a slight difference in timing. We don't yet know.
Okay, and also it seems very important, the growth rate of the clones. So do you think there is a plausible or practical way to determine the growth rate of the colonies in patients to know whether they are gonna develop faster or not?
That's an excellent question. I mean, my dream experiment would be to, take the healthy population, and we know that in the healthy population, maybe up to 1% of individuals have a low level of JAK2. And what would be really nice is maybe to longitudinally sample and see whether those who had a faster rate of increase in their aberrant cell fraction are perhaps the ones that then later developed MPN or later developed abnormalities in their blood counts or thrombosis. There are lots of healthy individual JAK2 mutated cohorts, and we are in early discussions as to whether there are some samples where one can do this. The trouble is we're talking decades of follow-up, and so the cohorts are tricky, but maybe retrospectively one can do that. One can look at the variant allele fractions in patients and try and predict perhaps were they the fast growers or the slow growers, but to do it in a larger population than we have done in the study. So I think the first step is to maybe do it retrospectively in a larger cohort and see is it still true, and then maybe to try and design prospective studies that involve healthy individuals with what we predict to be faster growing clones and see what's happening to their blood counts and their risk of thrombosis.
Okay, and to continue with you and then change, here there is a question that you mentioned that there is still driver mutations that have not been identified. Why is this?
Yes, we don't know. We had these clonal expansions in some of our elderly patients that don't have one of the canonical driver mutations. We're not the first to identify this. There's a paper by an Icelandic group many years ago at the time when clonal heme was discovered, published, I think in perhaps Blood, I'm not sure, it's Zink et al., where they did whole genome sequencing to identify clonal hematopoiesis, and they found that up to 50% of clonal hematopoiesis, you can find a clonal expansion, but there's no driver mutation. Whereas many of the original studies that reported clonal hematopoiesis are targeted sequencing. So you're looking specifically for certain mutations, you're not actually identifying novel drivers. So either there's something non-genetic, something epigenetic that's going on, or it may be that there are driver mutations that are not yet uncovered. And time will tell again, because this is an area of active interest for many groups, as to which of those possibilities it is.
Okay, and the last one, do you believe it's important to determine whether, for instance, DNMT3 mutation and JAK2 mutation are in the same clone or different clones? Because this is difficult for us in the clinical practice, in the lab to determine, but do you think this is relevant? Because this may affect the growth rate.
Exactly. Well, and we know from work that we've published that the order in which the mutations acquire, DNMT3A then JAK2, or JAK2 then DNMT3A, or indeed JAK2 and DNMT3A as separate clones, it influences the phenotype, it influences the selection. But sometimes I believe, because you can't tell in clinic, you just get a VAF of JAK2
That's
and
right.
a VAF of DNMT3A, sometimes you can't tell. What we do know is more mutations is bad, because more mutations usually is in elderly patients and it associates with poor prognosis. So in general, if you have more than one, it's a sign of you're getting older, you're getting more mutations. Specifically, I think it can sometimes explain the response to therapy, maybe. There is some emerging data coming up now that when you have interferon therapy, it can reduce the risk of the VAF of the JAK2 clone, but perhaps allow the DNMT3A clones to increase. I think it perhaps is interesting to know because it helps make sense in what's going on in the patient. But are we at a stage where we need to predict it going forward in patients? I think clinically, I find it hard to justify, and one would need to do these clonal assays. But I think it sometimes is useful to think about it because it helps make sense in terms of how the patient's behaving.
Okay, thank you very much. Now I have a question for Dr. Where is it? Here. DJ. So you have found these loci, these 17 or 25 loci with, that associate with higher risk of MPN. And have you studied these loci in families with MPN aggregation?
Yeah, it's a terrific question. So we're starting to look at a number of families where there's clearly multiple individuals who've had MPNs. We have two hypotheses that we're pursuing. One, as you've seen, is that these common variants, and we're starting to uncover more common variants as we expand these studies, likely have a very small impact individually. But the polygenic risk score derived from all of these variants in aggregate can actually have a big impact. And if you think about it, any two siblings will share about 50% or so of those common variants. And so it's very possible that you could get family aggregation when you have kind of extremes in the polygenic risk score. On the other hand, we are also very interested in trying to identify rare variants that have a much bigger effect size. And so we're doing a lot of studies, including with Jyoti and others, trying to identify these rare variants and trying to say, could those underlie what's seen in families? My suspicion is, especially as we've learned from other complex diseases and traits, is that predisposition is probably going to be some combination of those, of both of those, that there's going to be rare variants that we have yet to identify, but that likely have a big impact. And there's probably also going to be common variation in polygenic scores that are impacting people's risk.
So the people with higher risk with this genotype, risk genotype, if I understood well, it may do that the hematopoietic stem cell pool expands. They have like a higher number of hematopoietic stem cell pool, right? And then what happens is the driver mutation in these patients.
Yeah, I mean, I think it's an excellent question, and it's just a model at the moment, right? We know that there are a lot of people who have these germline variants who don't necessarily go on to develop myeloproliferative neoplasms. The question, of course, is, what's happening? Is it that, for example, having these mutations at baseline alters some property of hematopoiesis, at least based upon the studies we have done looking both in vivo at phenotypes, as well as trying to model some of these mutations. And I just gave you one example today, but in all those studies, it's just that baseline hematopoiesis might be altered. You could imagine that, consistent with what Jyoti was mentioning, perhaps there's also some impact of having these variants on the substrate of already having a JAK2 mutation. So, I think that in the future, it'll be very interesting as we're starting to get more insights into what the genetic variants are, how these might impact normal hematopoiesis, as well as how these might, in model systems, impact hematopoiesis in the setting of JAK2. So I think it'll be interesting to tease apart to what extent it might be predisposition to acquiring the mutation versus collaboration with a somatic mutation. And probably there's going to be some combination of both of those that's happening in these with these germline risk variants.
Yeah, because in fact we have families with several members with MPNs, and each of them carrying different mutations in different driver genes. So maybe that's why. And would you recommend to test an NGS panel with these loci in these families with MPN aggregation, or what? How would you act with these families? Would you approach a genome study with them?
Yeah, it's a terrific question, and I think
try to find the cause.
Yeah, it is a terrific question, and I think I do think we'll need a little bit more data than what we have right now. I think have a pretty good polygenic risk score, at least when we've tested in out of sample populations. But as we expand these studies, and already we're getting to the point where we have about 5,000 or so patients in aggregate. So I think we'll be able to develop better polygenic risk scores in the next, months. And our hope is to be able to, enable anyone who can do genotyping, and that can be done with low-pass genome sequencing or with genomic arrays, to basically be able to predict, what is the polygenic risk score for any patient. I think it's definitely worthwhile to look in those families for, are there extremes of the polygenic risk score that might underlie this? We know of cases, for example, with hypercholesterolemia and other complex traits where individuals can be at the extreme of polygenic risk score, and that can aggregate in families and show patterns similar to monogenic variants. Of course, at the same time, our hope is, as we are looking more at sequencing data, that there might be discoveries to be made in the rare variant space. And so hopefully, as those are reported, those might also underlie some of the cases of familial aggregation that are seen.
Okay, thank you very much. And at last, tengo una pregunta para Marta. Mi pregunta es, cuando habéis estudiado el perfil genómico de estos pacientes, ¿habéis encontrado diferencias entre los distintos tipos de síndromes en cuanto a cómo se distribuyen las mutaciones en los genes, por ejemplo, más mutados, TET2, DNMT3 y ASXL1? ¿Habéis visto diferencias entre los distintos tipos de patologías?
Sí, lo que pasa es que obviamente los pacientes que tienen mielofibrosis y tal suelen tener más mutaciones, y sí que es verdad que parece que los que tienen como menos expresión del cuadro hematológico, que caen en neoplasia mieloproliferativa inclasificable, suelen tener menos mutaciones. Lo que pasa que ahora nos gustaría también compararlo con los mismos pacientes, pero sin trombosis esplácnica, para acabar de verlo.
Ok. Bueno, pues me parece que no hay más preguntas. Congratulations to all of you for your amazing talks. Now ahora paso al siguiente bloque, and thank you very much to the audience for your attention and for the questions. I've learned a lot today. Paso al siguiente bloque, que sería el bloque clínico presentado por el doctor Hernández Rivas. Y muchísimas gracias por vuestra atención.